ಎರಡು ಕಾಣುತ್ತೆ ಅಂತ. Okay, we started the good afternoon. We started the class little early, like 3 minutes early. Not for anything. Like I just wanted to like tell things. Like some of you are saying to show the photos, right? Meiosis. Actually, we included one similar kind of an image in the initial step, and again adding 2 pictures. So in this particular 2 images, you can refer anyone. So as you can see, these are interface nucleus. So as you can see, this is like stagnant, right? No changes is happening in this particular nucleus. So it's almost stagnant, no changes or anything. But as you can see in these 2 things, later look at here, this interface nucleus is turning into some other things, like chromosome segregation is happening, and they are separating, right? And they are make it in an equatorial plate. I can draw it here, right? Equatorial plate. So metaphase and anaphase is like a speed up one. Okay, don't think that in this much quick only everything will happen. So this is the speed up photo. That's why it's like it's happening in like some seconds, because they are making it a GIF, right? That's why. And can you tell me what are these green color threads? These are like making a messy kind of structure and separation is happening. Can anybody tell me what are those? A green color mesh is something like you can see here, right? All these things. So a cotton like thread like structures. What are those, you guys think? Exactly. Like in some manner we can say spindle, but all these are cytoskeleton materials. Okay, cytoskeleton materials. Cytoskeleton material are having tubulin, actin, and myosin, different kinds of proteins. Okay, so that's what from there only this tubulin protein or actinomyosin is formed. This particular one, spindle fibers. As you can see here, remember, look, everywhere this cytoskeleton is completely filled the cell. But when cell cycle is running, just look through these particular position. Surrounding that particular cytoskeleton concentration or that particular protein concentration are comparatively low, right? Because what is happening? All those are redirected to inner side where actually spindle formation is happening, right? So redirection, rearrangement, everything is happening. You can like compare the things, right? Here also same thing is there. Once the cell cycle starts, it's become like blank, right? Why it's becoming blank? Because all of the cytoskeleton materials, some of these are like moved over for the another job, that is the formation of the spindle fibers. Okay, so good afternoon. So I just wanted to show these things one more time. As you can see here, itself one picture is there. Okay, this is also one of the beautiful picture. As you can see, chromosomes are moving away, but the resolutions are low. Sorry for that. Okay, because like this is GIF and everything, that's why resolution is comparatively low. Okay. Okay, guys, then we can move on then. I think everyone can hear me properly, right? So we will just see what we studied in the last class. So last class we majorly studied about mitosis. So what is mitosis? A kind of cell division which will give rise to 2 identical daughter cells. Right? So a single cell will be there. It will go through a step and it will produce the 2 cells which are normally identical. Probably now you understand why we name the first one as amitosis, right? So that's also giving 2 progeny only. That's also going through the division steps only, but it's not following the prophase, metaphase, anaphase, telophase stages. So that's why we called it as amitosis or binary fission. Okay. But if you check some books, okay, I'm not referring all the books, a very few book, I'm not sure like what are those books, also mentioned this prophase, metaphase, anaphase, telophase stages in the amitosis or in a binary fission, but don't go for that. That is not accurate. That is not correct. Okay, you can't differentiate prophase, metaphase, anaphase, telophase stage in the binary fission or in the amitosis. If in any book it's given like that, then it's wrong. Okay, so amitosis is a direct procedure. A single cell is there, it will give rise to 2. But in here, like all the steps are there, right? Prophase is like getting ready. That means the chromosome formation happens here, and the spindle fiber formation happens here. Like as this formation happens here, Metaphase can be represented by chromosome being in the equatorial plate, and in anaphase everything will be separated out, right? It's going away, separating out. And in a telophase, like it's already reached the point and it's again decondensating. Then last one we learned about the cytokinesis. Actually, we saw two types of cytokinesis, right? One in a plant and another one in the animal. Can anybody tell me what kind of cytokinesis happen in animal? Is it centrifugal or centrifugal? In animal cytokinesis is centripetal or centrifugal? Anybody? Exactly. Centripetal, right? And in the plants you can go for the centrifugal, because doubt in difference between spindle fiber and aster. Simple. All the asters are spindle fibers, but all the spindle fibers are not asters. So the spindle fiber in animal cells, we call it as a aster. That's it. Spindle in animal cell, we call that as the aster. Why we call it as the aster? Because it can produce the spindle in all direction. It looks like a star, right? So we discussed that. Yeah. So this much only we studied. I think everyone already like revised the things, so we can go today smoothly, right? I believed in that way. I'm not sure like how you guys did or what. Okay. So good afternoon, Gopika, Sanika, and here is also so many people are here. Okay, good afternoon. We'll come to the first question then. Okay, the first question is, which stage of cell division do the following figures A and B represent respectively? Okay, they are giving two figures and they are asking which are those particular cell division steps. Okay, cell figure A, figure B. Okay. So metaphase, telophase, later anaphase, prophase, telophase, metaphase, anaphase. Prophase and anaphase. Just compare this one and this one. Okay, this belongs to here, and this one belongs to here. Just take a look and select the option. This question is asked in 2010 NEET. Okay, so somebody asked, right? Do they also ask the diagrams? Yes, they do ask the diagram, but not very often. Okay, but in this chapter, it's normally they will ask. Okay. Okay, so I already launched the poll. Who are in the Zoom can answer in the poll, and who are in the YouTube can answer in the YouTube live chat. Like I'm waiting. This is one of the easy question, right? It's not at all hard. If you read a little bit, then it's not at all hard, right? If you don't know anything, then it will become difficult. If you don't know like a bit of a things also, then it will become difficult. So you have to choose in between any one, right? If you choose any one properly, then you can answer properly. Okay, still a lot of people having confusion. Last 10 seconds. You're not wasting any time. I'm ending the poll in 5 seconds. You have to answer quickly. I'm ending the poll. Okay, so I'm sharing the result. 13% choose option A, 24% choose option B, 58% C, and 7% D. Exactly. So what are the things you have to check? Where is the chromosome location, right? Chromosome is like, chromosome is somewhat like this. So what you can infer, chromosome you can visible, spindle fibers are there, this some nuclear membrane still there. Then they haven't given any interphase stage. That's why you can directly go as like prophase. Right. So if you check in here, prophase, only one answer is there. The options is correct, right? If you don't know about the prophase, leave it. Okay, go come here. Then what is the problem? It's not a metaphase because it's not arranged in equatorial plate, right? So it's not a prophase because it's already started separated. Then you may like having trouble with, is it a telophase or late anaphase, right? If it is a telophase, the cytokinesis already started, and this is already decondensation should have started, right? So you can go for the late anaphase. I told you, right? Understanding any one diagram is enough to answer this question, right? One of the easy question, I think, right? So any if you have any doubt regarding this particular question, just put it in the chat box. Okay, you can move on. Hi, Ashwin. So second question. Given below a schematic breakup of phases and stages of cell cycle, which one of the following is the correct indication of the stage or phase in the cell cycle? Okay, they marked something, they given a cell cycle picture. Okay, you have to see which one of the options are correct. C karyokinesis, D synthetic phase, A cytokinesis, B metaphase. Okay, I'm launching the poll. Do you guys need time? I don't think so, right? So I'm relaunching the poll. Okay, I launched. I gave almost like 10 seconds of time. So I'm waiting from both of you guys from YouTube and Zoom. I don't think it's a tough question, right? So, but don't get confused. So whatever hurry you are, you should like take a good look at the picture. Okay. If you decide something, sometimes that's what happens. Okay, you see something, then you'll get the answer. It will happen in like 2 to 3 seconds. Sometimes you have to go through all the options, but it will take almost like this kind of questions. If you know the cell cycle, it will hardly take 20 seconds, right? Hardly. That means 10 to 20 seconds. You should be quick because you have to save the time, because like physics and chemistry, you need a lot of things to do. You can save the time in biology only. You can't save time in physics and chemistry, obviously, right? So this is like a backup. So I'm ending the poll. It's already time is over. Okay, I'm ending the poll. And I'm sharing the results. 11% chosen option A, 60% B, 16% C, 13% D, and you need to be everyone choose an option B only. Okay, what is A is mitotic phase, and what is B? Here I can write prophase, metaphase, anaphase, and telophase. That means B is a telophase, sorry, anaphase. C is a cytokinesis, and D is the S phase, right? So next you have to connect this one. C karyokinesis, they given no wrong. D synthetic phase, they given yes, correct. So you can directly go here, but still we will see. A cytokinesis, wrong. B metaphase, the given. B is anaphase. So what's the correct answer then? The correct answer is option D. Okay. So don't get confused with these things. Okay, this ABCD and this ABCD, don't get confused like that. So this is different and this is different. Okay, don't do like that. Okay, so like read properly. So I think if you have any doubt, just post it. Okay, otherwise we can move on. So we completed this mitosis in last class. Today we are going for the meiosis. Okay, like why we need to study about meiosis? What's the function of meiosis then? Okay, the mitosis is always gives similar or identical kind of the cells, but identical is not that much healthy in the environment. Okay, because the environment is ever changing, then organism also have to change. Okay, have to change means there should be a recombination. That means a new combination should be there, right? And another one thing is like the mitosis always gives similar kind of the ploidy, right? If it is a mitosis, the 2n always give rise to 2n, and n always give rise to n. Remember, mitosis can happen in any kind of the cells. Okay, it may happen in haploid, diploid, triploid, tetraploid. Okay, nothing matters to that. Okay, it never matters the ploidy or anything. But if there is a chance you have to reduce the ploidy number, then mitosis can't do that. Okay, then for our rescue, we have another kind of a system. We call it as the meiosis. Okay, meiosis, what happens? We normally call meiosis as the reductional division, right? We already use this term, right? Reductional division. We use the term in like third chapter, I think, right? Third chapter, when we are discussing about the life cycles, right? I told you what happens. So in that 2n, the ploidy of 2n, that means two set of chromosome number will reduce into n. Okay, so that means what happens if I write this one chromosome like this, a homologous chromosome like this, then how this will form? It will form the 4 gametes. Okay, 4 gametes or 4 cells, each with a 1 chromosome or 1 chromatid, you can say, right? That means from the 2 chromosomes having 4 chromatid, you will get the 4 cells, right? This is a single cell. So what is happening then? here the reduction is happening, right? The reduction is happening from the parents to the half of the things. Okay, you may ask, sir, what's the need of that? Then why we have to go for the reduction? Okay, so sexual reproduction is very important. You already know, and you already studied a little bit in 10th, I think, right? 9th or 10th. So in the human reproduction chapter is also there, and reproduction general chapter is also there. You may once again study in the 12th class. Okay, the need of reproduction is getting a new chances. Okay, so getting more probability of good breed or good kind of the natures and everything. Okay, so for that we have to mix both the father and two species. Okay, two chromosomes have to we have to mix. We normally call those as the parents or father and mother. Okay, if we are using the this one, okay, mitosis as the process, then what happens? Father will give 2n, mother will give 2n, then that organism will have 4n. That means that organism may have 2 head, 4 legs, and in next generation what happens? 4n, it will become 8n, right? Tejas, I will come back to that. Okay, so this always happen in this way, it will go on increasing. That's not suitable, right? That's not at all suitable. That's why what happens, even though parent is 2n, the meiosis gives rise to n. Then it will combine, then again it will become 2n, the new progeny. Then it will be like maintaining the same chromosome number in 2 generations will happen, right? So that's why meiosis is very important. And one very good question, Tejas asked, does meiosis happens only in reproductional stage? No, it's not true, but mostly in reproductional stage, cells. Okay, but meiosis always linked with a reproduction. Okay, meiosis always linked with the reproduction. Why it happen in this way? Because if you in our second chapter, then we classify the meiosis, right? How the meiosis maybe types will be there. Okay, one is the gametic meiosis. Another one is the sporadic meiosis, and the third one is the zygotic meiosis. I don't know how many of you remember this one. Okay, I'm just repeating in a smaller version. Okay, types of meiosis. But this is not that much important, but understanding this is important. Okay, what's the meaning of gamete? Which is most abundant cycle. Exactly, most abundant. Really? No, mitosis is most. Okay. Gametic meiosis. Okay, so what is gametic meiosis? In this, what happens? In reproduction, meiosis only involved? No, in amoeba it's binary fission, right? Amoeba is binary fission. We are not talking about there when change in this one only. We will complete this one. Okay, then we will go to your queries. Otherwise, it will distract and everything will be something will happen. Okay, so amoeba is this one, binary fission. Okay, so remember here what happens? 2n parent will be there. Okay, this will give rise to the gametes, n gametes. So for this meiosis happening, that's why we call it as gametic meiosis, because it's give rise to the gamete. Okay, so this is what we normally see. This is what happens in our body to produce the sperm or the egg, everything. Okay. Second is a sporadic meiosis. What you guys saw in this one, pteridophyte and the bryophyte, right? There what happens? 2n parent is there, and this is giving rise to n, not a spore gamete. It's actually giving rise to spores, right? This is giving rise to spores. This spore will give rise to gametophyte, right? So that gametophyte will produce the gametes. It's a long process. I think you remember the plant kingdom chapter. So if it produces spores, so then we call it as a sporadic meiosis, right? And third one, zygotic meiosis, what we saw in the fungi and in some of the lower organisms, okay, lower algae. Here what happens? N from the one gamete and n from the another gamete, like parents, we can call it as parents. Parents, both of them are haploid. They are combining and forming a 2n zygote. But the issue is a parent is n, but the zygote is 2n. Then what is going? It's going through a meiotic process, and it's giving rise to the 4 individuals individual cells. Individual cell. So all of them are N only. Okay, N only. So, and all of them are going through the process of meiosis only. So in this process where it's happening in the zygote. So we call it as the zygotic meiosis. Okay. Try to understand. Wait 1 second. Okay. So what are the types of meiosis? The types of meiosis are gametic meiosis, sporadic meiosis, and zygotic meiosis. Gametic meiosis happens, will produce the gametes that normally happens. And sporadic meiosis can be found in lower organisms like lower plants and everywhere. So there actually diploid parent cell will give rise to haploid spores. In third one happen in very low kind of the organisms where haploid parent will give rise to diploid zygote. That diploid zygote again give rise to the through the meiosis haploid individual. We already seen this in plant kingdom. I just want to revise it. Okay. So where it happens then. Okay. The mitosis can happen in the somatic cells. Soma in the starting only we called, right? Where mitosis is happening, the somatic cells. So that means soma means body cells. Okay. Body cells, it will happen. Okay. So in the body cells, so soma means body. Remember that. Okay. We discussed it initially. But this meiosis happens in germinal cells. Germinal cells regularly. Okay. Germinal cells means what? Germinal cells means reproductive cells, or you can also simply say it as the cells which will give rise to the sperm or egg. Okay. Where it is present in human body, it is present in the testicles, in the male, and in the ovary in the female. Okay. So mitosis normally happens in somatic cells or in the body cells. Meiosis normally happens in the germinal cells or reproductive cells, which give rise to sperm or egg, which is situated in the ovary or in the testicles. Okay. So this meiosis is like a little longer phase. Okay. Give me 1 second. Yeah. Okay. So what happens? What we are discussing exactly. So this meiosis is a little longer. The interface is exactly same, no difference. Okay. But when you come here, when you come to the meiotic phase or M phase, okay, it's again divided into two types. Okay. So one is the M1 and another is the M2. Okay. M1 means meiosis 1. It have the prophase I, metaphase, anaphase, and telophase. Okay, and second one is again the repetition of step. Okay, why it is going through two types of steps, right? Within a meiosis, why we differentiated or why it's going through two kinds of the steps? Because it have to produce the haploid kind of the cells from the diploid. Okay, that's why it's going to double steps. We will learn about this. Okay, don't tension about prophase steps and everything. I will show one more time. Okay. So more widely happening process is mitosis. Absolutely. Okay. More widely happening process is mitosis, not the meiosis. Okay. Because wherever meiosis happening, that cell has to come from the mitosis only. That's why. But what you have to remember, the meiosis will give rise to 4 cells with a reductional chromosome number. Okay. That's why the meiosis happens only in the diploid cells. Okay. Only in diploid cells or above that. Not in a haploid cells, because haploid cells can't go any further division, right? That's why. Can we observe it lively? Exactly, you can observe it. Once you come back to the school, no, like for 11th and 12th, meiosis live observation is not, live observation take a lot of time. Okay, the secondary, but like section is not there, but already made slides are there. You just have to see the, go through the slides only. Okay. So we will move on to this one. So just an overview, guys, how it will happen and everything. Okay. So as you can see, meiosis 1 and meiosis 2 is here. In meiosis 1, the single cell passing from the interface will give rise to the 2 cells. Okay. With a reduction of chromosome number, we will see how it will happen. In meiosis 2, that 2 cells will give rise to additional 2 cells. Right. So simple. In meiosis, 1 cell will give rise to 2 cells. In meiosis 2, 2 cells will give rise to 4 cells. Okay, simple maths, simple calculation. 1 to 2, 2 to 4. Okay, you have to remember this much only. Then we will go through a little depth. Okay, so First phase, so what we are studying is the prophase. Okay, prophase is longest phase in this one also. Like in the particular steps, so this also comparatively little longer. Okay, not 100% longer. It's comparatively longer. Okay, so this prophase we can again divide into some other steps. Okay, like why we did that, right? So in that it's easy. One step we can complete it in 2 slides, but here we again divided 5 times, because the prophase in mitosis and prophase in meiosis is different. Remember, prophase in mitosis is like direct. Okay, it's just condensation is happening, and the nuclear membrane dissolving happening, nucleolus dissolution is happening. Okay, but here that won't happen. Like that will happen, but along with that some other steps also happen. Okay, so what are those then? The steps are named after the different kinds of the methods they used. Okay, so we keep it in that way only: leptotene, zygotene, pachytene, diplotene, and diakinesis. Okay, yeah, so remember leptotene. Yeah, I told you, right? Interface will be there in, it's a common interface is common in mitosis and mitosis and meiosis. That's what we written here, right? Interface is common, no difference in the both of the steps. We are only discussing the meiosis. Interface is exactly same. That's why we are not discussing it again. Okay, no difference in interface. Okay, so that's why we directly came into the M phase. Okay, so leptotene, zygotene, pachytene, diplotene, diakinesis. As you can see here, something is happening. We will see in detail how it will happen. So you can also remember by with another one, like different kinds of making mnemonics or anything. I normally prepare like lazy people will die soon. Like I simply thought this one. Okay, how it will happen? Like the first one, like lazy, L for leptotene and Z for zygotene, and this P for pachytene, and di only is there. Di means diplotene and diakinesis. That means di. Diplotene you can remember in between. Okay, so lazy people will die soon. So that's why don't be lazy. So you can remember in that way if you're having trouble understanding or trouble remembering this particular one. Okay, so we will go in detail about each of these phases. The first phase is the leptotene phase. Okay, so why I'm having not lot of diagrams, because a lot of the diagrams are not accurate or not good. That's why I left with my hand, like hand drawing only. Okay, so bear with that. So what happens in this particular step? The chromosomes become distinct. Remember, wherever I am writing in the prophase, think that this is the nuclear membrane. Okay, think that this is the nuclear membrane, and outside the cell wall will be there, and the cell plasma membrane, anything will be there. We are not writing it. Okay, the nuclear membrane, you have to remember this. What's happening in here? The condensation is started. Okay, but not 100% done. Okay, because 100% done in the metaphase, you already know that, right? Condensation is started, and this become distinct. The chromosomes will become distinct. Okay, that means you can directly visible, and they are longest and thinnest. Why it is longest and thinnest? Because condensation is not done. Okay, if it is condensed, then it will be shorter, right, and thicker. But condensation is not done. That's why it's longer and thinner. Okay, so and on chromosome contain a series of beaded structure appear called chromomere. Okay, I will write two chromosomes like this. Okay, if you observe clearly, you can see a beaded kind of structure in between. Okay, this beaded structure we call it as the chromomere. So you may ask, so what's the chromomere? Okay, so chromomere, we are not 100% sure. Okay, but chromomere, some people will say that these will represent the functional gene region. Okay, the region where actually functional genes are there, right? Somebody will say that, okay, these are those regions. Okay, but we are not 100% sure yet. Remember, okay, when you are talking about these kind of things, there are several things will be there still needed exploration. Okay, someone want to like go for like scientific field as a researcher or something, can take up these kind of things. Okay, can work on all this. Exactly. Okay. So these are called as the chromomere. Okay. And here the centrioles divides into two. Remember, not centromere. Centrioles are dividing into two. Centrioles are here and it's divided into two cells. Okay. So that's why it can go make a centrosome. Okay. And the ends of all chromosomes are directed towards the centriole. Okay, what's happening? The ends of the chromosomes are directed towards the centriole on one side of the nucleus, forming a bouquet. Okay, what is the bouquet? You already know meaning of bouquet, right? Bouquet means like this, like some flower bouquet. We normally call it, right? Flower bouquet. So how it's appear? This is also appears somewhat like that, right? Because everything, if I write more chromosome in this manner, then it will looks like a bouquet. Okay, why it look like bouquet? Because the chromosomes, the ends of chromosomes are directed towards the centriole. Okay, centriole. That's why it's like almost like looking like bouquet kind of the shape. Okay, so that's what we call it as the leptotene or the first stage. Okay, so these are the small stages. Any doubt regarding leptotene stage? Any doubt regarding leptotene stage? Or if you don't understand any sentence or anything? So you have like always have yes or no options. Like go to participants. Okay. If you think we can move on, click on yes. Otherwise, click on no. If you think I have to slow down, click on go slower. If you think I have to go faster, you can click on go faster. Okay. So use those options. So second step is the Jagotin step. Okay, don't I think that I haven't added any diagrams. I simply left it because I didn't find any good diagrams. Okay. In first three slides, I didn't understand, Chirant, what you are saying. That one. So all the threes are there. Yeah, got it. Two homologous chromosomes approach each other and being a pair, this pairing is called synapsis. Okay, remember what is happening here. So one homologous chromosome I can return, one chromosome I can return like this, and another one chromosome I can return like this. Okay. In G1 phase is longest in meiosis also. That will be like no, like that will be like we are not completely okay. Here another one phase is the longest. We will come to know the change. Okay. What's the difference between chromosome in first three chromosomes and in last two? First three chromosome and last two means I didn't understand. What is first two and last two? This slide, first three, bro, like I'm for a model I wrote down. Okay, no tension. Everything is same only. Everything is chromosomes only. Okay, somehow beta structure. I forgot to add it beta structure because I wrote it later on. Don't take tension about this only. Don't take tension about this slide. I'm telling you later. You guys will come to know that. Okay, don't take tension about that. Okay. So, remember what is happening this one. So this one, sorry. Okay. So this one is from the parent, or how can I say? Sorry. Okay. This one is from the father and this one from the mother. Okay. So this is how you can call it as the homologous chromosomes, right? These homologous chromosomes approach each other. Sorry, I should not write like this. I have to write somewhat like this. Okay. Because the chromatids are not that much visible in this stage also. Okay, so these are come near and approach each other being a pair. Okay, we call this pair as the synapsis. Okay, what's the meaning of synapsis? Synapsis means pair of homologous chromosome. Okay, pair of homologous chromosome. Okay, and each pair consists of a maternal and paternal chromosomes, right? That's why we can also call this pair as the bivalent. Bivalent. The gap between two nerves is also synapses. Exactly. Okay, that synapse is different and this synapse is different, but everything means in a close contact. Okay, the synapse means in a close contact. The meaning is similar, but don't confuse with that to this. Yeah, okay, correct. That's also correct. Okay. So while teaching this in our sir also told the same thing. Okay, our sir told that don't confuse with that synapse. So that synapse is different and this synapse is different. Like, okay. So when you guys asked that, I just remembered that one only when I'm in BSc. Yeah. So each pair, what is happening here? These are two things are there, right? That's why we call it as a bivalent. Two chromosomes are here. Okay. And in this particular stage, the chromosomes are becoming more thicker and shorter. Okay. Remember, throughout the prophase, the continuation is happening. Condensation is happening. So every one step further, the condensation is more, right? Up to the metaphase. Okay. And in between, what happens? They develop a structure. Okay. In between, they will develop a structure. This particular structure is made up of the protein. Okay. This protein structure, we call it as the synaptonymal complex. Okay. Synaptonymal complex. Synaptonymal complex. What are these structures? These are the, why they are not have the chromatid? The chromatid is there, but you can't differentiate the chromatids because condensation is not over. Okay, condensation is not over. That's why you can't differentiate the chromatid. It is there only. Okay, so so in between the two homologous chromosomes, we can find a synaptonymal complex or a protein complex. Okay, what it is doing then? It is keeping both the chromosomes in a tight contact, both the paternal and maternal chromosomes in a very tight contact. Okay, and while all this is happening, the centrioles will move to the opposite poles. Okay, centriole will start to move to the opposite poles. Okay, we are still in the prophase only, remember about that. Okay, so any doubt regarding anything in this particular one, in this particular stage? What is the difference between bivalent and synapses? Bivalent simply means two homologous chromosomes. Okay, synapses means those are in a pair. Okay, so similar kind of the meaning, but like different meanings for the same thing. Okay, different meanings for the same thing. Okay. Any doubts? Otherwise, click on yes, so I can move on to the next slide. Okay. So third one is a Pachytene stage. Okay. Fourth point. Okay. I will come back 1 second. I told you, right? I told you this is 1 second. I told you, right? This is the nuclear membrane. I told you I am only writing the nuclear membrane, not the cell membrane. I told you in the initial only. That's why. Okay. Fourth point. That's what only. Okay. In between the homologous chromosome, a protein structure will develop. That's what we call it as the synaptonymal complex. Okay. Synaptonymal complex will what happens? It will composed of DNA and protein. It will make two paternal and maternal chromosome in a close contact. Okay, this is a nuclear membrane. Don't this one. The outer cell membrane is here. I'm not writing it. Okay, space is not there. Next one is the Pachytene stage. Okay, so here. Remember, this is the nuclear membrane. I will write down. Okay, that's the better thing. Nuclear membrane. So what happens here? You can differentiate the chromosomes. Okay, so, sorry, I'm using this color. Sorry for that. I will use white and yellow only. Okay, so here you can see the chromosomes with the chromatids, right? So what happened here? Condensation is more pronounced. Okay, more condensation has happened. That's why you can see the chromatids. So we call this one as the tetrad. Okay, tetrad. In that you probably remember dyad, right? In a single homologous chromosome, you can find two chromatids. We call it as the dyad. Here, two homologous chromosomes, and they are jointed also. and they're having 4 chromatid, we call it as the tetrad. Tetra means 4, right? So 4 chromatid you can find, okay? And due to the increased attraction, what is happening in between and due to this nocturnal complex, these are becoming highly coiled. These are becoming highly coiled, okay? And these non-sister chromatids in this particular structure, remember, always the non-sister chromatids, that means this one with this one, are going through a recombination, okay, or going through the recombination. Then what's the meaning of recombination? Okay, so recombination means sharing the part of the DNA, okay, sharing the part of the DNA. If you observe this particular diagram, here I can see blue chromatid here and red chromatid is here. What is happening? They are actually changing, or they are lying one above the another, okay? That's what we call it as a recombination nodule, okay? Nodule means a bead-like structure, right? So it looks like a bead-like structure wherever the crossing over is happening. So this particular step, we call it as the crossing over, okay? Crossing over, wherever crossing over is happening, a bead-like structure will appear, okay? Don't confuse this bead with the Leptotene beads. That is different and this is different, okay? So when it's like placed one on another, there are like 2 enzymes will work, okay? The first one enzyme which work in a recombination is same as the name says, recombinase, okay? Recombinase. What this recombinase enzyme will do? This recombinase enzyme will transfer some of this DNA to the another chromatid and this chromatid part of this chromatid to the other chromatid. As you can see, no, where I told the centrosome is inside, centrosome is outside only. I haven't written the centrosome. Like, don't confuse. This is nuclear membrane only. Never centrosome will come inside the nuclear membrane. Never, ever, ever. Okay. So remember, this is nuclear membrane. Centrosome I haven't even written. Okay. These are the tetrads. These are the chromosomes. Okay. What is happening here? So crossing over is happening. That means they are sharing, right? Their DNA sharing is happening. That's what we call it as the crossing over. Okay. So where it will happen, this crossing over only happen with a homologous chromosome. Okay, homologous chromosome means what? Homologous chromosome means from the 2 different ends or 2 different origins, right? But they should be same. That means father's first chromosome will always go for crossing over with mother's first chromosome. Okay. Father second always go with the mother second. Father's third always go with the mother's third, because the size and everything matters there. Okay. So changing is happening, crossing over is happening. Okay. And when crossing over is happening, that means cutting everything done by recombinase, then we have to join it, right? For the join, we have the another one enzyme, we call it as the ligase. Okay. What this ligase will do? Ligase will reunite this part. Okay. Reunite this part. Because of that, what will happen? Crossing over formation will happen. Crossing over function will happen, or formation will happen. Okay. I'm repeating everything once again. Here, the chromosome become more condensed. So you can clearly see the chromatid. Okay. Each chromatid. So we call it as the tetrad stage. Okay. Because 4 chromatids are there. And what happens along this synoptonemal complex? This non-sister chromatids, what they are doing, they are going for the pairing. Okay, they are going for the pairings. And pairing means what? They are crossing each other, and there's an enzyme called recombinase. What it will do, it will take some part of the leg of the one chromatid and another chromatid, and it will exchange the DNA. Okay, that process, exchanging, is called as crossing over. Okay. So after the crossing over, when the DNA is like already like changed, right? The DNA should be stitched back, right? That work can be done by an enzyme called ligase. Ligase will exchange or join the part or reunite the part. Okay, we will see. Nishchay, once again. Okay, it will join the parts, and by the end will be looking like that. So you have to remember one important thing. What is that? the crossing over always happens with the non-sister chromatids, always with a non-sister, not with a sister. Don't think that this will cross over with this one will cross over with this one. That never happens. Why it is not happening? Because if it happens also, nothing will like useful will come, right? Because both of them are identical. That's why the crossing over always happens with non-sister chromatid. You think this one as the like humans, okay, where marriage will happen. Think recombination or crossing over is like a marriage, okay. So where marriage will happen? Marriage will happen always happens with the other persons, right? Not with the family members, right? Sisters will never get married with a sister, right? Or brothers, right? Exactly same kind. It always, the crossing over is also exactly like our family system. It always happening with the non-sister chromatids, not with a sister chromatid. Okay, understood. So we will move on. So next stage is the diplotene. Okay, so in last stage crossing over happened, okay, but you can't normally differentiate that because here the synaptonomal complex is still there. Okay, synaptonomal complex is still there, but that's why it looks like jointed only. But when diplotene stage come, then the synaptonomal complex are started to broke down. Okay, synaptonomal complex are started to broke down. So I will write down again nuclear membrane. Okay, so what happens here? Here, these chromatids or chromosomes wanted to go away, okay, wanted to go away, but this synaptonomal complex, sorry, this crossing over is not over yet, okay, not over. That's why what happens to this, in this particular structure, these are going in this direction, these chromosomes want to go in this direction, but the issue is like in between they are jointed, okay, in between they are linked. That particular linkage will look like X kind of structure or a kind of X, okay. That's why that structure we normally call it as the chiasmata, okay, chiasmata, okay. So that exchanging area where gene exchange or DNA exchanging is happening, that's what we call it as the chiasmata. It's become visible, okay. I will ask one question here, okay. So I told, okay, chiasmata will started to visible and everything. Can anybody tell me where chiasmata formation happened? In which stage the chiasmata formation happened? Can anybody tell me? Simple. I'm asking in which stage chiasmata formation happened. I'm not asking prophase, dude. I'm asking about the stage in the prophase. Which stage? Which stage? Exactly. Remember, the chiasmata formation happened in the Pachytene stage only, but it is not visible that time. Why it is not visible? Because synaptonomal complex is there. Both of them are intact. You don't know where it's connected or not. In the diplotene stage, you will start to see the chiasmata. That's why I only mentioned chiasmata become visible, right? I haven't mentioned like it's formed here. I mentioned chiasmata become visible because it will formed in the Pachytene stage, but it will be visible in the diplotene stage. Okay, and this is going away, right? Repelling stage. That's why we call it as the desynapsis. That means ulta to the other stage. Okay, that stage synapsis is happening, right, in the zygote stage, but diplotene is a complete ulta. Desynapsis is happening. Okay, diplotene is the some of the longest stage in some cells. Okay, not in all cells. In some of the cells, it may take 12 to 15 years. Okay, like we can give the example of human female that's also in the egg, or instead of saying egg, it's not an egg actually. Oocyte, I can say it's a oocyte. Okay, the oocytes which is present in the human female ovaries are normally in the diplotene stage. Okay, normally in the diplotene stage. When that particular girl reaches to the maturity, then they will move on to the diplotene stage, okay, and they will complete the meiosis process and they will give rise to one egg for like every 1 month. Okay, that's what happens. Every 1 month it will release one egg, but until then it will remain in the Diplotene stage. So that's why if you consider that one, that will be the longest kind of the phase. Okay. So, you will see in detail in the 12th class, okay, how it will happen, why it will happen, everything. But you have to remember, one girl child is born, she already contain all her eggs. Probably you already read about it in 10th, right? When you are reading about human reproduction, right? So all the eggs she can produce throughout her life, everything will be already present in the ovary, in this kind of the stages, okay, in between stages. Okay, so that's what happens. So if I sum up the diplotene is ulta, that means desynapsis will happen. That's why the chromosomes start to move away. Because of that, we can see X-shaped structure in between the homologous chromosomes, where DNA exchanges happened. That's what we call it as the chiasmata. Okay, X shape. I will write like here also, X shape. Okay, so they asked a question about chiasmata. That's why I mentioned that. Okay, so as you can see here, right, X shape. So last one is the diakinesis. Okay, diakinesis is like easy one. So here, remember, now I'm writing this cell membrane. Okay, cell membrane, and this is the nuclear membrane. Okay, here already the reached pole, this asters are already reached pole. Okay, here if I write this one, chromosomes like this, sorry for taking extra time. Okay, yeah, correct Ashwan. I thought of mentioning it here because some people will get confused that way. Okay, good, correct, absolutely correct. Okay, so what is happening in here? Okay, terminalization is completed in this stage. So what is the meaning of terminalization? Okay, so as you guys see here, right, probably in this one, we saw the chiasmata formation has happened. So once the chiasmata formation has happened, this chiasmata will move away because it should get that both of the chromosomes should be separated, right? That's what initiate the terminalization here. Okay, initiate terminalization. Terminalization means getting separated. Terminalization means getting separated. Okay, they are getting separated. It will start in here in the diplotene, but the terminalization will be complete. The procedure will be completed in the diakinesis phase. Okay, the bivalents tend to repel each other and migrate to the periphery of the nucleus, just inside the nuclear membrane. Okay, nuclear membrane are like little bit intact, and this nucleolus will start to disappear, and nuclear membrane also start to disappear, and spindle fibers are start to appear. Okay, but if you see after the effect, after this prophase, this paternal chromosome having some of the part of the maternal chromosome, and this maternal chromosome has some part of the paternal chromosome, right? So this is very important. The prophase one gives rise to what kind of the chromosomes? It will give rise to the recombinant chromosomes. Recombinant means what? Recombinant means new combination happened. Okay. Late joining because another class was there. Venkat, you. So what happened? The chromosomes, they are getting separated, and while they are going, what happened? They are taking some part of the other chromosome. So it gives rise to a new set of chromosomes. Like this chromosome is different from the what chromosome we initially had, right? Because that chromosome is pure. One is faternal end, one is the mother end. But here, these are recombinated chromosome. But here also just observe one thing. What's that you have to observe? Only the one set of chromatid is actually recombinated. Another is exactly parent type, right? This one is the parent type only, and this one is also chromatid parent type only. But these two are recombination happen, right? Recombination, where this happen, recombinated. Okay, recombinated. So no, terminalization is same as crossing over. No, terminalization means ulta of crossing over. There actually crossing over is happening, right? Here the crossing over is removing. Okay, removal of the crossing over. Okay, removal of the crossing over. Crossing over not visible. Please use other colors. Where? Here. Here crossing over is done. Here is there is no crossing over in this one. The crossing over is done. It's already separated. Okay, it's separated. It's an individual one. It's an individual one. It's separated. Okay. So this is no longer in a like joining. This is no longer holding hands. Okay, these are no longer holding the hands. They are already left the hands. Okay, so they are like again become like feeling like we are individual. Okay, so what we seen then in from there to here. Okay, if you summarize it, we will go to the first slide. Now you guys can understand here, right? In the leptotene stage, this becomes segregation is happening, right? Segregation is happening, or condensation is happening. And in zygotene stage, what happening? Synaptonemal complex formation is happening. That means they are joining. Look, this chromosome is joining with this one because these are quantitatively same, right? The length is same. This one is joining with this one, and this one is joining with this one. All of them are bivalent, or all of them are homologous. You can't join this one to this one because size is different. It won't happen. Okay. Next, Pachytene stage, crossing over is happening, as you can see here, right? Pachytene crossing over is happening here. And in Diplotene stage, because they are going away, you can see the chiasmata formation or X-shape formation, right? And in the Diakinesis, they already separated. Okay, they are already separated. They showed already separated one, but they merged it a little bit. Okay. So what's the use of recombination? Recombination will give new combination, right? Recombination is giving a new combination. If new combination happened, then father chromosome will remain like a father chromosome only, mothers will remain like a mothers only. But because of this recombination, what is happening? Character mixing, right? You are getting the mixed character. You are getting the mixed character. So that's the important. Getting a mixed character, getting a new combination. That's the important. Okay, importance of that particular one. Any doubt regarding any phases of the prophase? You have the time, you can ask. So otherwise click on yes. Okay, if you have any doubt, just put a message. Okay. Good afternoon. Okay then. So we in first 3 slides, chromosomes are parallel. In 3-4, it's like X-shaped. Exactly. In first 3 slides, in this slide or in this slide, what happened is like this complete condensation is not over. That's why this is not properly visible. The chromatids are not properly visible. But from the Pachytene, the condensation is almost more increased. So you can differentiate the chromatid. That's why it's X-shaped. Okay. Before that, the condensation is not that much. That's why it's almost like a single thread-like structure you can see. Okay. So next is the metaphase. From here, like it's easy, but this metaphase is very important. Okay, because it's different than the meiosis. So here, all 3 types of microtubules are well developed. What are those 3 different types of the microtubules? If you ask me, so you already know. One is the can anybody tell me what are the 3 different types of microtubules? If you don't know, just put like don't know. Okay, no issue. It is like this only in NCERT given. No, mitosis they even haven't given this one. Meiosis they given this one. Mitosis they given different picture. Exact. One is a continuous. Exactly. One is a continuous spindle fibers. Another one is the kinetochore spindle finder, which are attached to the kinetochore, and another one is in between. That will be interjonal. Exactly, Akshay. Right. Kirtana and Yashwant. Exactly. Correct. Absolutely correct answers. Okay. So continuous, that means not connected to the chromosome, or kinetochore spindle fiber, which connected to the kinetochore. Interjonal, that means between the chromosomes. Okay. So all the 3 types are well developed, all the 3 types of microtubules, and the chromosomes move towards the equator, and finally they orient themselves on the equator. Remember how they are arrangement is happening, or something happened. sir. Okay. It went to Zoom. So how they are arranging then? They are arranging as the homologous pair, right? They are arranging on the pair, homologous pair. Remember, this is very important. Okay. Homologous pair. As you can see here, this is one chromosome and this one is the another chromosome. Okay. Meiosis, the chromosome are from parent only. Grandparent, we can't say like that. I will come to that later. Okay, last we will discuss how it will happen. Okay, Tejas. But for now, it's only parents only. Okay, we won't confuse in that manner because it's a little confusing. Okay, we need a lot of imagination power to that. That's why. So as you can see here, like homologous chromosomes are arranging on the equatorial plate, not the single chromosomes. Two chromosomes are arranging. Okay, and they are arranging in a random manner. Don't think that fathers will always arrange on one side, mothers always arrange on one side. Okay, fusion is not over. Exactly, fusion is not. Fusion is over already. That crossing over, everything is over. Here they are arranging only. They are arranging only. Crossing over, everything is done. Okay, but they are arranging. Don't think that father's chromosome will take one side, mother's. No, nothing will happen. It will arrange randomly. Look here, in these two chromosomes, red is this side, but in this one, red is this side, right? It's completely randomly arranging, but homologous are arranging properly. Okay, and these chromosomes move towards the equator and finally they orient, and this centromeres lies towards the poles and the arms towards the equator because they are pulling is happening. Okay, this at this particular stage the chromosomes have the two chromatids, exactly two chromatids. You can clearly differentiate it, and these chromatids are highly condensed one. Okay, highly condensed one. So what's happening? The only difference between metaphase in mitosis and metaphase in meiosis is metaphase in mitosis, only one chromosome will lay on the equatorial plate, right, on one angle. But here, the homologous chromosomes, that means two chromosomes or a pair, will be arranging on the equatorial plate. Okay, so we will see the anaphase, then you will understand properly. Okay, then anaphase 1, what happens? The chromosomes contraction, that means pulling and pushing will happen, and that will help the chromosome to move in a direction. Remember, here a single complete chromosome is moving one direction, this complete chromosome is moving another direction. Already their recombination is done, as you can see here, right? Recombination is done. They are having some other chromosome pairs also. Because of this, a complete chromosome is moving away. That means one centromere is going in this direction and another centromere is going in this direction. In this particular stage, the cell become diploid to the haploid, right? Because up to it it's diploid because having two centromere, a set of chromosomes. Here one centromere move in this direction, one in this direction. That's why it's almost become reduction or haploid. Okay, we will see. So I will read out one more time. What's happening here? The segregation will happen or disjunction will happen. Okay, remember wherever you heard the term disjunction or segregation, go to anaphase. Okay, so it's moving in the two direction, and the division of centromere is absent. Here centromeres not at all division is happening. If you go for anaphase of the mitosis, there it happens, but here not. Because why it is not happening? Because total chromosomes are moving, right? The total chromosomes are moving, not divided ones. Understand? So total chromosomes only moving to the two different directions. Okay. So here centromere not at all splitting. That's why I mentioned like two sentences here. Okay. When you have the time, like when you complete this class, read it properly. Here centromere does not split. Okay. So only the complete chromosome, one homologous chromosome going in one direction, one pole, another homologous chromosome going in another direction. So this will reduce the number of the centromere. That's exactly equals to the reduction in the chromosome number. So in this stage only, the diploid will become haploid. Okay, diploid will become the haploid. Okay. So next one is the telophase. Telophase is simple. Exactly. Okay. The haploid number of chromosomes, as soon as they reach the pole, what happens? They will start to decondense. Okay. And they start to uncoil. Okay. The nuclear membranes start to reappear and the daughter nucleus start to form the everything. Okay. This will happen. Okay. So next one is the cytokinesis. The cytokinesis is a little different here. Okay. So how it will different? After meiosis 1, the cytokinesis only completes in animals. That means once it's done, thus animals only form 2 cells. Okay. But in plants, this cytokinesis not happens here, but it will happen once second meiosis also over. Okay. So here it will give 2 cells, right? Plant cells don't do that. Plant cells will remain like that. Once all the meiosis will done, it's directly give rise to 4 cells. Okay, but here the animal cells will give rise to 2 cells because of the cytokinesis. Okay, and here is also when plant cell directly give rise to 4 cells. Okay, directly give rise to 4 cells. We call those 4 cells as the tetrad only. Okay, so don't confuse this tetrad with the other tetrad, chromosomal tetrad. Here tetrad means 4 cell. There tetrad means 4 chromatids. Okay. Yeah, this is the telophase stage 1. Okay. And what happens then once cytokinesis is over? It has to start the second prophase, right? Before it will start the second prophase, a short duration time will be there. Okay. A short duration time will be there. We call that short duration time as the interkinesis. Okay. Don't think that it's an interface. It's an interkinesis. Okay. Kinesis means karyokinesis means what? Division, right? Inter means in between. A gap in between the 2 division. A gap in between the 2 division. That much only. Here, no interface will occur. Nothing will happen. The interkinesis, a short duration will be there. Okay. It is not also obligatory. Okay. What's the meaning of obligatory? Obligatory means it should be there, right? But this interkinesis is not an obligatory stage. Okay, that means wherever you are observing that, you should not say that, okay, interkinesis should be there, right? It's not possible. Interkinesis may be there, interkinesis may not be there. Okay, in that interkinesis, nothing will happen. Okay, the cytokinesis will continue or it will start. This interkinesis only lasts for a few minutes. Okay, lasts for a few minutes, a few seconds to few minutes only. Okay, so no changes will happen there. Okay, normally no changes will happen. Okay, normally, because this exceptions, if you take every stage, like different things will happen. Okay, that's why. So one question came. So after this question, we will go for some other things. Okay, dissolution of synoptimal complex occurs during. They're asking dissolution of synoptimal complex occurs during. Okay. Pachytene, Diakinesis, Diplotene, Leptotene. I'm launching the poll. You can answer in the poll. Who are in the Zoom, who are in the YouTube, can answer in the YouTube live chat. Okay. So actually, I'm checking one more time. Last year, from this chapter, 4 questions are asked. Okay, in a NEET, 4 questions asked. That means 16 marks. Okay, 16 marks. If you made it correct, that means it's almost equals to the like 20 marks. Okay, 16 to 20. That means like it can change the ranking in thousands. Okay, thousands your ranking may be vary. Okay, so that much important this chapter is. Even though this chapter is small and this chapter is easy, this chapter is very important. Okay, remember 16 marks is not a simple thing. Okay, 16 marks. So 4 questions actually. 2 questions from meiosis and 2 questions from meiosis. So 2 questions from the interface. I'm ending the poll. Okay, I'm ending the poll. I don't know like how much time happened. More than one. 25% chosen option A, 30% chosen option B, 41% chosen option C, 5% D, exactly. And YouTube also everyone chosen option C only. Simple. So what they're asking dissolution of synoptimal complex. Okay, that means what? That means desynapsis. Where we mentioned the desynapsis word, we mentioned the desynapsis word in the diplotene stage. So that's why the correct answer is diplotene. Simple, direct, done. Okay. So That's it. So direct kind of the question, right? If you write that particular last desynapsis word, you could have answered this. So we will move on. So we will move to the meiosis 2. Okay. So before moving on to the meiosis 2, if you have any doubt regarding meiosis 1, just put it in the chat box. Or if you think you don't have any doubts, then click on yes. Okay. So I think then we can move on. If you have any doubt, you are free to put it in the chat box. Okay. So what is happening in here, meiosis 2? In meiosis 2, exactly same procedure is happening, but like in a more quick way. Okay. So we normally call meiosis 2. Okay. Anaphase one more time. Yeah, I will go back. Sorry. Anaphase is simple. Don't confuse about all these wordings. I added all these things only for your reference. Okay. What happens in anaphase? The homologous chromosome will go on to the different pole. Okay. I will write it in a simple lining here only. Okay. Homologous chromosome move on to different pole. Move to different pole. Okay, different pole. So that's what happens here. There is no splitting of centromere, nothing, no segregation, this one. Okay, separation of chromatid, nothing will happen. One homologous chromosome go to another one pole, and another one go to the another one pole. Okay, exactly. That's why what happens, because of chromosome moving to the different poles, centromere number decreases, right? That's why reduction will happen in the number of chromosomes. Okay, number of ploidy. So this one, right? So meiosis 2, we call it normally call it as the mitosis kind of the division only. Okay, mitosis kind of the division. That means equational division. Okay, why we call meiosis 2 as the mitosis? Because it's exactly like a mitosis. Okay, there is no difference in the meiosis 2. You can see here. Okay, so prophase 1. In prophase 1, what happens? The chromosomes. One second. Sorry. Okay. It's again started something. So what happens here in the prophase 1? The chromosome will again appear distinct. I will write something like this only. Okay. I will write. I think that it's a plant. Okay. Or we will keep in this way only. No issue. Okay. So what happens here? The centrioles again divide. Okay. Centrioles again divide. The nuclear membrane will disappear, and the chromosomes are again start to become more and more condensed. Okay. More and more condensed. But remember, two different things in the two different one, right? So how it will happen then? Here is the recombinated, and here is the recombinated one. Okay, so each entry will start to divide, and it will start to go on to the poles, different poles, and they start to produce the esters. Okay, they start to produce the ester. Nuclear membrane is already dissolution is happening. Okay, and nucleolus will disappear. Nuclear membrane is also disappear. Simple as in the metaphase. Okay, prophase 2. Important is the metaphase 2. Okay, metaphase stage, what happens? In this, as you can see here, I will write a cell in this manner. Okay, cell in this manner, one cell in this manner, and another one cell in this manner. So here, so if I write this one, centrosomes like this, okay, this chromosomes will arrange themselves. Chromosomes will arrange themselves. Okay, don't confuse about in between writing. I'm just making the writing proper. Okay, that means it have to show the like this, right? So what happens? They will arrange in such a way that towards the pole, centromere, and everything. Okay, as you can see here, so arranged in a equatorial manner. Remember, first division happened, first equatorial happened in this manner, right? Because chromosomes arranged in this plane and they got separated. Now it's happening in this manner. So try to arrange the this one alignments. Okay, how it's happening and everything. So it's arranging in this manner. Okay, it's arranging in this particular manner. So one second, guys. So I simply texted someone because chat box is not working. Okay, Akshay, I simply texted, don't take attention. So this is what happened. So this is arranging in a new manner or in a new segregation. So as you can see in the metaphase, if I give the single cell, you can't differentiate, right? Apart from this recombination. So it exactly like metaphase cell, metaphase of the mitosis. But the issue is like mitosis metaphase, only one cell will be there. Here, two cells will be there. Okay, here two cells will be there. What about the telophase then? The two chromatids of each chromosomes are separated by the division of the centromere. So this is where the good things started to happen. Okay, so I'm again writing the same cells. Okay, writing the same cells, and I'm taking the centrosome here, centrosome here. Okay. Homologous chromosomes are not there, but here how it's moving means which color I used there, correct, right? So it is moving in this manner. One second, give me 1 second to write down this diagrams. Okay. Exactly. Okay, so this is how it's happening. We are not writing all these things. Okay, remember how it is happening in here. Then the chromosomes are moving into the different poles. Chromatids are moving into different poles. How it happened here? The anaphase is exactly like mitosis. Exactly like mitosis. That means what is happening here? That means the centromere splitting is happening. The centromere splitting is happening. As you can observe in the last metaphase, I'm going to the last slide one more time. Okay, sorry, it ended. Here, as you can see, metaphase is, the chromosomes are intact, right? Both the chromatids are intact. But what happened here? In this particular structure, the centromere is already separated. Because of division of centromeres, the chromatids are moving on to the different direction. Okay, moving on to the different direction. And what is happening? The contraction of spindle fiber is happening, and it's actually taking this centromere away. Okay, now you guys tell me. Okay, before that only it's n, and it's n only because of only one ploidy, one centromere. Now, is it any reduction is happening in the chromosome number? No, chromosome number reduction is not happening. Why it is not happening? I will write down here. Because in this one, a single chromosome, cell is like this, then it's giving rise to the two cells with chromatids like this or chromosome like this. Here, if I saw a red dot, a single centromere, okay, a single centromere is here. If you check here, a single centromere only here, right? A single centromere, and here is also a single centromere. That means here is also n, here is also n, and here is also n. That means equational division is happening. Equational division is happening. That means separation is happening, but chromosome number is not getting reduced. Okay, so that's why leave us to the telophase stage. So this is one of the easy phase. What happens here? The daughter chromosomes again go for chromatin thread. That means the decondensation is happening once again. Decondensation is happening. Okay, and nuclear membrane start to appear. Okay, nuclear membrane is started to appear, as you can see in here. Okay, and nucleolus start to appear, and spindle fibers going to disappear, exactly like the metaphase telophase. Okay, so this leads to the last cytokinesis. If it is like animal cells, as you can see here, like this, first one give rise to two cells, which again give rise to four cells. But if it is a plant cells, then a single cell will be there. This single cell only, every division will happen. This division will give rise to four nucleus structure. This will again go for like this kind of things don't happen, right? This will start to produce this cell plate formation in all the direction, and this will give rise to the four different cells. Everything is n in nature. Okay, n in nature or haploid in nature. Okay, so cell plate happening. This is the cell plate. Okay, this is the cell plate. If it is the animal, it's giving rise to 2, and that 2 will give rise to the 4. Okay, so that is what happened after the cytokinesis, end of the cytokinesis. So any doubt regarding the meiosis 2? Any doubt regarding the meiosis 2? So if you didn't understand any particular stage or anything, we will like glance one more time, right? That will be better. Glance will be better. So here, as you can see, two cells are there, and these two cells are what they are doing. These two cells are giving rise to the two cells. Here, it's again going for the prophase. In prophase, no crossing over, nothing is happening in here. Exactly like mitosis, right? So these are arranging on a plate, and this plate, the chromatin threads, this spindle fibers are coming and attaching it. Okay, and what's happening in this particular stage, anaphase? Here, centromere breaking is happening, and they are going away. And telophase, again, decondensation is happening. Good question, Chayant. What's the difference between anaphase of 1 and anaphase of the 2? In the anaphase of 1, the homologous chromosomes are moving away, right? And centromeres are not at all dividing. But in anaphase 2, the centromere is getting divided and the sister chromatids are moving away. Okay, sister chromatids are moving away. Remember, in anaphase 1, homologous chromosomes are moving away. In anaphase 2, sister chromatids are moving away. Okay, that's the this difference. Okay, that's the difference. So what is the significance then of the meiosis? So important is the formation of the gametes. They will produce the sperm and the egg, right? They will produce the sperm and they will produce the egg. That's the very important one. And they carry the genetic information. That's important from father and to the, instead of, I can say like parent to the offspring, right? Parents to the offspring, they will carry the information, and they maintain the chromosome number. That means n to the n or 2n to the 2n, right? I think you understand how it's n to n and 2n to 2n. If you don't understand, go to the first slide where we discussed about different types of the meiosis, right there. And they will help in the assortment of the chromosome. That means fathers in fathers only and mothers in mothers only, right? That's very important. And they help in crossing over. That means changing. Okay, they changing what is happening. That will allow that. And they won't cause the mutation. They will carry the mutation, right? Carry the mutation. The mutation may be good or bad. So if it is good, it will be good, right? If it is bad, it's bad, right? Exactly. So if a mother have a good mutation, then it will carry to the offspring. It should, we are not saying that it should be in any one parent. It can be in any parent, right? Like, so we made a difference. So difference is simple. So in mitosis where it occurs, mitosis occurs in somatic cells, but meiosis occurs in the germ cells. Mitosis nucleus divides only once, but meiosis it will divide two. I will come back to the children once again. Mitosis gives rise to 2 daughter cells, but meiosis will give rise to the 4 daughter cells. And mitosis daughter cells are diploid or haploid, depends upon the parent, okay, but meiosis is always haploid. And mitosis it occurs frequently, okay, but more frequently, but meiosis less frequent, only in the germinal cells, okay. And mitosis happens in somatic organ, but meiosis gives rise to gametes, okay. Mitosis have 1 stages in everything, but meiosis 2 stages. And mitosis chromosomes are not at all changing, but meiosis, the chromosome number is changing, okay? And the chromosome number doubles at the beginning of each cell division, but in between it not doubles, it only doubles in the initial, right? So no crossing over, but here crossing over can be seen, and it's an equational division and it's a reductional division, okay? In further gametes, how maternal chromosomes are present? Then it's the chromosome of grandparents take role of the parent. Yeah, says, yeah. So third option, maintenance of the chromosome. I told right, maintenance chromosome. If you want to understand, in starting I mentioned about zygotic meiosis, right? Just look into that, how the zygotic meiosis will work. Okay, there you will get the answer. Good question, Tejas. Remember, I will write something like this. Okay. About the grandfather to fathers. Think about 4 people, okay? Think about 4 people and 2 male and 2 females, okay? One male is having this in his sperm and the female have this one is her egg, and another one person have this one in his sperm, and another one person having this one in her egg. Okay, male, female. Okay, when both of them are combined, then that particular person will have this particular one, right? Sorry, not this one. This particular chromosome set. I think that this is your father. Okay, this is your father, and another person will have something like this, something like that. Think that this is the male, this is the female, this is the father, and this is the mother, right? So now you are coming here, gametes or production is happening. How the gametes may be, right? The gametes may be different, right? It may be directly from any one of the father. Like I'm writing the 4 gametes in here. Okay, I'm writing all the 4 gametes in here. 1, 2, 3, 4, and 1, 2, 3, 4. All the possible 4 gametes from your father and all the 4 possible gametes from your mother, right? How it may be. So one gamete may be exactly like your grandfather, right, without any changes, and another may be a recombinated one. I simply left it like that. Okay, leave the crossing over in the first step, no issue. So one is exactly like another, like you are from other chromosome. Okay, sorry, grandmothers, but another one is the crossing over. Remember, every step crossing over is happening. I just omitted in 1 or 2. Okay, same thing happen in here, right? One is from the exactly like your this one, like your grandparent, and another one exactly from another one grandparent, right? So you are a combination of any one of this, right? You are the combination of any one of these 4 to these 4 combinations. This can be anything. This can be something like this one to this one, or this one to this one. It's like completely different, right? So you guys make some chart like this, then you will understand. Okay, here I omitted the crossing over, right? Here I completely omitted the crossing over. I just simply took it like it's exactly coming without crossing over. You make a crossing over, then you will come to know that how many combination will happen. Okay, that's why there may be possible from the grandfather to the you directly a chromosome may came, but you have the 46 chromosomes. All the 46 coming is a probability is very less, right? Very less. So that's what. So this leaves this question, this particular chapter. If you have any doubt, you can put that. Okay, if you have any doubt, you can discuss or put that now. So I will discuss that. Before next, we will start the morphology. Okay, in the next class, we will start the morphology. Before morphology, I will also add 2 more slides. There I will explain about how the DNA and the chromosome number will change along the mitosis and meiosis. Okay, for that to done, you have to complete mitosis and meiosis. Okay, that to done, you have to complete the mitosis and meiosis. So homework is read mitosis and meiosis properly. Okay, then only I can explain how the DNA and chromosome number will vary along the cycle. Okay, that's why I'm not mentioning that today. Okay, so I'm leaving it there. Venkat, I think you are happy, right? You are very happy, I think. So we are starting the morphology chapter. Thank you. Just don't forget, okay? Have to read mitosis and meiosis and complete chapter. Complete chapter. Read and also solve the module. This is your homework. What do you guys think? Shall we go to photosynthesis or shall we go to morphology? Shall we go to plant physiology or shall we go to morphology? What do you guys think? So, everything based on new plant physiology, yeah. Morphology. Okay, no, don't take tension. Like we will complete morphology and anatomy, then we are going to the plant physiology, because some points in morphology and anatomy are needed to understand the plant physiology. Okay, that's why. Good, Shushma. Right. Once we complete the plant physiology, we are coming back for the structural organization, and then we are going for the animal physiology, because the things which you learned in structural organization are important for the animal physiology. If I complete in the first place only, then it will be problematic for the you won't remember. I know you won't remember until I will go to the animal physiology. Okay, human physiology. No chapter is deleted for you guys. So don't think that the chapter is deleted, that won't come in NEET, because you are not writing NEET this year. You are writing the NEET next year. So none of the chapters are deleted for you guys for the NEET next year. So don't ever think about like that. You have to read all the chapters in all the subjects. Okay. So remember, we are also doing. We are not at all leaving any deleted chapters. Yeah.